Welcome, everybody. We'll give just a few minutes to let everybody log in and kind of settle in, and we'll get started around 12:02 or so. All right, well, we'll go ahead and get started. Welcome, everybody, to July's iteration of the Frontiers Research and Discovery Grand Rounds. My name is Jamie Foup. I'm the Senior Director of Clinical Research Operations here at KUMC. I also co-lead the Frontiers Translational Endeavors CORE. So excited to be joining you all today. The Research and Discovery Grand Rounds series is a multitude or one of many resources and events that Frontiers hold. So we definitely encourage you to continue to go on to the Frontiers website at frontiersctsi.org, register to be a member, take a look at all the resources that we can provide you. By becoming a member, you get access to those resources and notices of upcoming events. And then, speaking of Frontiers events, I want to let you know that next month on August 22nd, same time, we'll be joining on Zoom to go over our next Research and Discovery Grand Rounds for Transforming Healthcare, the Path to Learning Healthcare Community, presented by John Spiritus and Stacy Farr. So look forward to having you at that point. With that, I definitely am excited to introduce our speakers. We've got KUMC's Andy Godwin, Chancellor's Distinguished Chair in the Biomedical Science, Professor and Director of Molecular Oncology, Division Director of Genomic Diagnostics, and then the Director of the Kansas Institute for Precision Medicine, COBRE. And he's joined by his colleague, Hirsch Pethek, Research Associate Professor in Pathology and Laboratory Medicine, Director of the Biomedical Discovery Lab, and Co-Director of the Kansas Institute Precision COBRE Biobank and Biomarker Validation course. So super excited to hear about their institute and their core services. With that, I'll hand it over to you. Thank you guys so much for joining.
Well, thank you, Jamie, for the introduction, and Ed Etherbeck for inviting us to present kind of an update on our Kansas Institute for Precision Medicine phase 1 and 2 COBRE. And so it's going to be a bit of a tag team where Hirsch Pethek, who has been with me for many years. He was with me at the Fox Chase Cancer Center in Philadelphia and then came to KU with me in 2010 and has established a lot of the laboratory activities that he'll talk about today. But I'm going to give you a little bit of a background towards what we're doing. I do have conflict of interest, but nothing affecting this talk, and Hirsch has nothing to disclose. We're going to talk a little bit about, I'll give an overview of the Precision Medicine Institute that is being funded by an NIH COBRE grant. And then we'll talk briefly, I'll do a brief overview of some of the core research cores and other resources. And then Hirsch is going to give some examples of support that we're doing at research here and other places in regards to the biobanking and biomarker validation core. So I think everybody's pretty much familiar with the field of precision medicine. I mean, it continually grows. When I got involved in this, we didn't even have a name for it. It's kind of called translational research. And so I've been in this field for more than 30 years. And really, when we started out, it kind of became known as kind of molecular medicine, genomic medicine, individualized medicine. And really, it's the basis of, advances in science and technology that lead to a better understanding of our health at the disease and molecular level. Even though my focus has primarily been in the area of cancer, there are growing areas across all of medicine where precision medicine will really affect how we're taking care of patients, either through diagnosis or treatment. And so what my work basically for most of my career has really been looking at the ideas of trying to link the clinical outcome information with molecular data in my case, for many years has been profiling tumors. And as part of the Cancer Genome Atlas project, I've part of a number of other NCI-driven studies characterizing tumors and trying to link that to clinical outcomes. But really it is kind of the goal is to develop these future disease classifiers. And it can go across, as I mentioned, multiple different kinds of diseases. So when I came to KU in 2010, I had an interest in trying to develop a variety of different things. And I've built many different kind of programs since I've been here, including molecular oncology for the first time. So we developed labs to actually do testing of patient samples to guide their care. But I also went to, at that time, EBC Samari and asked him about kind of developing an activity around precision medicine, because I knew it eventually could grow and influence a lot of different areas of fields within our medical center. And at that time, we raised some money to kind of launch the activity. And then shortly after that, there was an RFA released for LOIs looking to select the next COBRE application. And that was back in August of 2016. We put in an LOI with Steve Soper, who's on the KU Lawrence campus, who's a Foundation Distinguished Professor who had recently arrived, and we had been kind of working in similar spaces and we're kind of linked together, put in an LOI. It was reviewed as identified as being potentially of interest, but it suggests that we also work with Alan Yu, had put in one through the Kidney Institute. And so at that time, we kind of joined forces between Steve, Alan, and myself. But we're then told we had to wait to be considered because they were going to put in a second competitive, it had been a second application, A1, for a tumor microenvironment. So we waited until, again, the RFA went out in May of 2017. We were then selected in July 2017, wrote a grant that went in January 2018. And then you kind of can look at all this phases of how long it actually takes from going from the concept of 2016 to actually getting funded in 2019. At that time, this was our group. We had put in 5 research project leads. We had 3 research cores and then 1 resource, which is our patient and community engagement resource, which is kind of unique to most COBREs. And so that's one of the things that we continually are touted about is the really integration of patients' voices within the research that we're doing within our program. But when the grant actually got awarded, we only had 2 research project leads who were either here or eligible. So that required us to go and identify some additional ones, and that what we did with 3 additional people we added to our program. And this really was the very first 5 research project leads that were funded. And again, each of the research project leads get about $150,000 over 3 years to help build up some of the research program. With the phase 2, we're now funded 10 research project leads. We've been able to, as shown kind of in the award, we've been able to get $750,000 in supplemental awards for our phase one, and we recently got a $250,000 supplemental award, which is not included in this number here for our phase two. And again, our cores and resources remain the same. Just to give you an idea of the breadth of the research we support, the first five research project leads research span the areas from autoimmune arthritis to cystic fibrosis to implementation medicine approaches, as well as studies of identifying biomarkers using mathematical models, and then a study of head and neck. We added to that a supplement in Alzheimer's disease and then funded two pilot awards, which are at the level of $50,000 for breast cancer and colorectal cancer. We then awarded again, research project leads in the area of minimal residual disease and breast cancer, autoantibodies, and asthma. You can see here the smaller ones are pilot awards, protein radiotherapy, extracellular vesicle trafficking. Then again, the larger ones, the RPLs, we did something in radiogenomics in head and neck tumors. We did something in schizophrenia and sleep disorders. And then you can see again, a variety of different kinds of ones, from thrombocytopenia to role of platelets in cancer, to T-cell immunology, to again, minimal residual disease, and a different one, which is plasma cell disorders. And then most recently, we added our in the phase two, another research project lead focused on looking at the microbiome in ovarian cancer. So when we look at really the metrics, and this is some of the things we have to follow and we'll also always report because that's what you get reviewed on. We've done okay in the areas of our granting, but we've done very well in the areas of presentations and publications by our project leads. As primary PIs, we've seen about 9.5 million in return on investments in some of those, and they're part of more than $30 million of grants as co-investigators as well. Our project, our pilot program project, really was funded through institutional funds that I was able to acquire. And you can see again there, we've given out at this point a total of $500,000 in pilots, and we've seen a return investment of 3.6 million already, and 42 related publications. So when you look at the overall metrics of that, we've had 346 publications associated with our COBRE to date. And you can see that it's highly influenced by some of our cores, the BBV core, the omics core, as well as the BME core. And so you can see that our cores are very active, as well as our pilot project leaders and our research project leaders. So it's been very productive in that regard. There have been some studies, and one recent study that came out that really was looking at what are the true impact of these COBREs. Are they really doing what they're supposed to be doing? And that's kind of accelerating research within institutes within states that are relatively underfunded and federal dollars. And so we started to kind of compare some of how we're doing relative to the other 90 COBREs. We were not part of this review at the time, but they reviewed 90 phase 1-2 COBREs. And in general, when we put in our phase two, we were very competitive in regards to how successful our RPLs were relative to other COBREs. If you look at the area of publications, we're absolutely, at one of at the top, even as a phase one, just leading into our phase two now, where we have 346 unique publications. You can kind of see where that falls across all these other 90 phase 1 and 2 COBREs. And again, we always diligently work with our RPLs in regards to getting independent funding. And we've seen some improvements, but in the areas of federal funding, it's still a bit of a struggle. But we've seen areas of other foundation fundings, DoD-related fundings, pharma fundings, because something that's a little different from ours, other COBREs, is we have a high emphasis on physician scientists. So most of the, more than 60% of the ones that we have supported are physician scientists who have clinical practices as well. Something I literally have been working on is that we're looking to get a support from our Kansas Board of Regents. They have a program that they'll provide matching funds for certain kinds of grants. And so when we did that, I kind of looked at some of the metrics where our Kansas Institute for Precision Medicine actually spans 13 academic departments, 4 schools, and 6 research centers and institutes at KU and the medical center. We're supporting 15 companies in Kansas across the nation through our core labs. And one of our co-PIs, which is Steve Soper, started Biofluidica, which is housed in the Innovation Park on the Lawrence campus. And we also have seen our investigators filing patents for some of the discoveries they've done, as well as that there have been 3 startup companies. So we're pretty active in the areas of influencing some of the activities within the state, which is important for demonstrating to try to get these matching of funds. Our phase 2 looks like this, and I've mentioned most of the people going forward. The biggest difference was I was able to get them to change the policy regarding the advisory committee, because they wanted only 3 external advisors. We had 5 previously. I got them to waive that, and we've been able to add back on 2 of our external advisory board members. And so this now makes up our advisory committee. We no longer have internal advisory committee. But one of the aspects that I wanted to mention before, again, was our patient and community engagement, and that's led by Hope Crable and then patient advocate Cheryl Jernigan, along with Tanya Yelder. And one of the things we were able to do out of the patient and community engagement, along with philanthropic money I was able to do, was to start the Center for Genetic Services and Health Equity. And really, this is an effort to provide education and care to marginalized populations in our region. So we provide free genetic services for individuals from families at higher risk for inherited cancer syndromes who otherwise have no access to these. These are our stakeholders, current stakeholders that really contribute to this. Juntos has been a huge driver, along with Hope Crable and the Masonic Cancer Alliance. And these really are our community partners, and it really is a community. It's a KU Medical Center community activity, because all of our patients are all our participants become patients within KC Care Health Center. And so we've seen really a good impact of what we've done. We offer, as I mentioned before, free genetic screening, counseling, and testing. We've hosted many events in person to get the word out. Much of that has been led by Juntos, who, again, we've done a lot of work with some of the Spanish radio stations. Overall, within the Hispanic community, we primarily serve about 60% are Spanish speaking, 69% Hispanic. And most importantly, which I want to point out, is almost 60% are uninsured. These are individuals who had no idea that they would be able to find out if they had an increased risk or not. And as you can see here, those are receiving genetic testing results in the way that they're being positive. We actually have more men than women, which was a little bit of a surprise when we put this data together. But what really was important about this activity is that in order for us to do this, and we had a lot of pushback, and it took us several years to get this really up and running, is that anybody who is tested positive, we have to find a home for them. We can't just tell them they're positive. So we've really worked very hard, again, through the leadership of Mariana and Teresa and Hope to identify ways that we can refer patients who are found to be positive for a mutation in a gene that increases their lifetime risk of cancer. So insured ones, we can get some of those into the health system. Uninsured and underinsured, which represents more than 60%, we've worked with a number of organizations who will provide support for them for follow-ups and screenings, and those are shown here. And recently, Show Me Healthy Women in Missouri actually have posted one of our videos we've created in order to provide education in regards to how they can get access to this free services. They posted this right on the front page of the Show Me Healthy Women website. So I'm going to be turning this over to Hirsch, but I'm going to quickly just go over the fact that we do have research cores. They're open to anybody within basically our institutes, whether it's KU, the KU Medical Center, Kansas State. outside. We do a lot of work with outside groups as well. These are the three primary research cores that we started in basically 2018. And Hirsch will talk a little bit about the BBV core activity. I'm not going to talk much about the quantitative omics core, but that's led by Devin Kessler. And again, that's supports many. You can see there's many publications that have come out of there from supporting a number of studies through research project leads, pilot projects, and others. And then the biomedical engineering core, I'm just going to mention that briefly. It's run by Maggie Wittek as well as Steve Soper. And there are two components to this, and this really is kind of a partnership between a center grant that we have, which is called CBM Squared Precision Medicine, and that's Steve Soper is the director, and I'm the associate director for the P41 Center. And we are able to merge the resources between the Kansas Institute for Precision Medicine and CBM Squared to offer some of these services around plastic engineering as well as liquid biopsies. And the plastic engineering units are over on the Lawrence campus. It's actually a fairly robust area that they can do to actually custom design microfluidic chips for individuals who are interested in working with them, and they can prototype, or they can actually, take people's designs and manufacture them. We also have a liquid biopsy unit that's on this campus. It's a few floors down from me in Wall Hall West. Wall Hall East, sorry, that will has the ability to look at both circulating cells, whether they're tumor or normal cell types. They can look at the extracellular vesicles as well as viral particles, and then also look at circulating DNAs and RNA molecules. So there's automation that was built, and this is part of what I mentioned before with Biofluidica. That's the platform that we use. And we have several of those units here that are supporting a variety of clinical trials. And I think at last count, between the BME and the BBV core, we've supported more than 30 clinical trials, looking at different biomarkers and evaluating how those can be used to help assess response, whether it's prognostic or predictive biomarkers. And we actually have a pretty wide range that we've been able to serve throughout the United States. A lot obviously are centralized within the Kansas, Missouri area and the campuses that are there. But through the CBM Squared and the Kansas Institute for Precision Medicine, we've been able to support a number of different studies for the last, this is over the last couple of years. And with that, I'm going to pass over to Hirsch to talk about some of the specifics that he's doing, leading the biobanking biomarker validation course. So Hirsch, I'll let you take over.
Okay, thank you, Andy. So I help Dr. Godwin run the molecular biology arm of the BVV core. And this core is run by the individuals shown here. They've been with the core anywhere from 2 years all the way up to 6 years. They have a lot more experience even before they joined the core, as you can see by the variety of techniques that are shown here for each of the individuals. And this is just a brief listing of what they have talents in. So next, please. So the BVV core helps support faculty members here at KMC as well as outside of KMC with a variety of studies, including support for nucleic acid related studies. We help support with a variety of isolations from bodily fluids, conditioned media from cell cultures, as well as extracellular vesicles. Next, please. We also provide services for targeted DNA sequencing using custom panels that may not be offered through other genomics core facilities on campus. There is a QIAGEN interpret translational software that helps report pathogenic variants that might be detected from these targeted analyses. And we also help run microRNA, small microRNA sequencing studies as well. The core also supports typical mRNA and microRNA expression studies. We have two or three different platforms for that. One would be just a typical qPCR BioRad platform. Another one is the microfluidic Fluidigm Biomarker platform, which allows you to do a 96 by 96 multiplex qPCR reaction to help reduce costs as well as the input requirements for your DNA for qPCR experiments. And then we also have a direct count hybridization system, the nanostring encounter system, that allows you to do these mRNA and microRNA expression studies without the need for a traditional PCR-based amplification. So this is the encounter system is basically direct hybridization. And then finally, our newest addition to the core, which was added a couple years ago, is the nanostring geomics digital spatial profiler platform, which allows you to do, in this case, transcriptomic analysis on fixed tissue samples. So a single slide of your tissue will allow you to get expression data on 1,800 cancer-related transcripts, or approximately 18,000 whole genome transcripts for human assays. And then for the mouse-based assays, they have a 21,000 whole genome transcript assay as well. For protein-based assays, the core has support for typical ELISAs that you would do with a plate reader from a variety of biological fluids as well as conditioned media. We also have the BioRad Bioplex system, which is basically your Luminex system for bead-based multiplexing of marker analyses. And okay, next please. And we recently added a Zeiss AxioScan high throughput slide scanner, which allows you to do up to 100 slides per run. It can do both bright field as well as fluorescent based scanning, and it is capable of doing up to 40x full scans of your slides, allowing you to go back and visualize the slides with their software. We also have the West system is a capillary-based Western blotting platform. We don't have this system in our core itself, but it is available as a department instrument that we can help run, investigators help run this West capillary Western blotting system. And it can be used for cell lysis as well as extracellular vascular analyses. And then finally, coming back to the geomics DSP system, in addition to the transcriptomic assays that I mentioned earlier, it is also capable of doing proteomic analysis on your unstained FFPE slide sections. So for protein-based assays, you can see there's a smaller panel of 87 immuno-oncology-based proteins, as well as 76 proteins for neuroscience-based studies. They also have similar panels for mouse IO and neuro panels as well. And more recently, they've come out with a 572 protein panel for human protein analysis. It's not available for mouse at this point. Earlier this year, the core added the IsoPlexis IsoSpark instrument. This instrument allows you to do single cell proteomic analysis on a limited number of panels that are shown here on the right, as well as for your traditional bulk analysis of serum, conditioned media, and other biological fluids. The advantage for this system, it is fully automated. Once you load these single cell or bulk chips that are shown here, the machine will automatically run the collections and analysis, and the next day you would have your results. Their built-in software does much of the post-collection analysis for publication-ready figures. The core does also provide services for isolation and characterization of extracellular vesicles. We use a variety of methodologies just depending on the project that the investigator is interested in. Using traditional ultracentrifugation to isolate extracellular vesicles, as well as size exclusion chromatography. And we work with the BME core for immunocapture-based extracellular vesicle isolations. Next, please. Following EV isolations, we do support initial characterization of extracellular vesicles using nanoparticle tracking analysis or protein surface analysis using the ExoView platform, as well as the capillary Western blot analysis, the WEST system that I mentioned earlier. All these three platforms are not particularly in our core, but they are available on campus, and we do help investigators run these instruments as needed. And then finally, imaging through the imaging core facility on campus, we do help with visualizing the isolated extracellular vesicles using negative staining and TEM imaging. Next, please. We do have a pipeline worked out with the other core facilities that are part of the COBRE, specifically the BME core. We do work with the BME core for the immuno isolation of extracellular vesicles based on a specific immunocapture marker that an investigator might be interested in analyzing. Once EVs are immunocaptured, we do microRNA isolation, microRNA sequencing, and a preliminary data analysis on the microRNA sequencing data to provide investigators with initial data analysis on differentially expressed microRNAs within their samples. For additional, more complicated analysis, we work with the omics core for more downstream analyses that the investigator may be interested in. Next, please. And then finally, for the last few slides, I want to focus on the digital spatial profiling studies that we've been supporting on campus using the NanoString GeoMix instrument. So this is a very basic workflow that is shown here for the DSP studies. Basically, a formalin-fixed unstained slide is needed. This gets, we work with the investigator to identify optimal morphology markers that will help identify cell-type specific cell-specific markers within their tissue. These then get processed through the instrument, where the instrument will collect only from those cells within a region of interest that's designated, that we help work with the investigators to designate on the tissue. You can collect up to 3 different cell types within a region of interest based on your morphology markers. And those cell type specific collections will then get processed through the instrument for either transcriptomic or proteomic analysis, depending on what the interests are. And then again, there's preliminary data analysis on differentially expressed transcripts or protein markers that are identified within those regions of interest. Next, please. And the next basically slides are showing some representative studies that we've supported with investigators on campus as well as on campus. And so we've worked with a variety of individuals, investigators here, as well as outside, to perform transcriptomic-based and proteomic-based analyses on human tissue samples. And you can see from these, there's almost any tissue that's been collected has been studied, it seems, from through our core. The point I want to make from here is that it varies from cancer transcriptome to whole genome transcriptome analyses that are shown here, or proteomic analyses. Many of these are either in the process of being published or have been published. we've already published with Dr. Birkeland, a protein-based analysis on skin samples. We published with Dr. Saeed on cancer transcriptomic-based analyses from a Camilla phase 1, phase 2 trial that was conducted here, and are doing subsequent analyses for additional samples from that clinical trial. And then finally, next slide, please. We can do similar analyses for mouse-based studies as well. Again, the mouse are limited to whole genome transcripts, and then the smaller protein panels, they don't have the larger protein panel at this point. And then I think I will just a couple things that before you go to Andy, I went over the molecular biology arm of the BVV core. What I did not add on this is the biobanking component of the BVV core, and that's basically the bio specimen repository core facility on campus, which has provided many of these tissue samples to us, either working with the investigators directly and processing them, or these have been part of clinical trials that have been processed and provided through the repository core facility, as well as serum and plasma samples that we've used for a lot of our extracellular vesicle studies as well. And then, Andy, I think I'll let you go over the next slide.
Okay, and so, again, we've supported a lot of studies in-house, and Hirsch mentioned we've done a lot of clinical trials. This is an example that we recently did. It was reported in June at the ASCO meeting in an oral presentation based on a clinical trial through the Southwest Oncology Group. So I'm the vice chair of the breast committee's translational medicine group. And we had a concept there of going back and evaluating this study that was recently reported called S1007, that really was the second largest study that looked at the 21 gene panel, which is the Oncotype DX, and then looking at patients that had hormone positive, HER2 negative, node positive breast cancer and their response to adjuvant therapy. Well, what we found from that is that there wasn't a great separation at times between self-reported menopausal status, those that are pre and post-menopausal. So we proposed to obtain the samples from RxPonder and evaluate these for 6 serum markers. That's why I have this listed as old biomarkers repurposed. And what we found from this was that one marker that we had suggested, which is the serum anti-Mullerian hormone, AMH, was a much better indicator of adjuvant chemotherapy benefit than reported what they currently use, which is menopausal status, age, and potentially other serum hormones like ER and PR, and a few others. And so what we found is that the ovarian function, which is the measure of this, because most of the time people think of AMH when they're looking at fertility, we did it based on assessing whether ovarian function would be a good predictor of response, and indeed we found this. And so this is a potentially practice-changing discovery, and we're working on moving that into a CLIA lab here, because there's only one other lab in the country, a clinical lab in the country, that offers this specific test. Most have the fertility testing set up, and that is not the same as the ELISA-based assay that we basically applied and developed in our biomarker discovery lab and biobanking and biomarker validation core. And this is really now going to, I believe, change clinical practice, where it can affect thousands, if not tens of thousands of women's decisions each year about having adjuvant chemotherapy or not. So with that, I just wanted to give you an example of a national study that we've recently done, and now we're expanding this, which we just recently did to a French cohort, and we're analyzing that data now. And then hopefully we know that people who have seen this presentation are already ordering this test, but they're not ordering the correct test. And so we're trying to trying to educate the oncology population or breast oncologist about what tests to use and what not, because we've basically identified a specific level that would suggest that they would benefit from chemotherapy or not. So with that, I think we stayed on time. Yeah, we did pretty well. I'm happy to take any questions if there's any that are that people have. I will advertise right now that we have 2 pilot grants that are available for people to write an application to. I don't remember exactly the due date. It's sometime in August. We sent out a notice right before the 4th of July. And I think it's this first or second week of August when the grants will be due. And then we'll have 2 additional pilots this year as well. One that will be cancer focused, and that'll be supported by the Cancer Center. And then we'll have another one for pathology faculty. And so we'll have a total of 4 new pilots this year, and we hope to do that for the remaining 4 additional years. So we're hoping to be able to do as many as 20 pilot grants over the course of this phase 2. And then if we're successful with our COBRE application, we will also be advertising a 5th research project lead position. So stay tuned for that. So with that, I will stop sharing and see if there's any questions. Anybody's posted in the chat? I don't think so. I don't see anything in the chat. Oh, we got a kudos. Yes, thank you, Lisa. Yeah, they have done a wonderful job. They're great to work with. And most of what I do in the area of fundraising seems to be trying to get money for juntos. So if there are any questions, you can raise your hand or just unmute. We're fine with that if anybody has, or any comments you want. Okay. I don't see any.
Well, we really want to thank you for presenting today. That was extremely intriguing and great science and community engagement going on there. So excited to learn how things go. Definitely, if you have any follow-up questions, please reach out to Frontiers or Ashley Carlson or myself. We'll get those directed. Otherwise, we'll be excited to see everyone next month, August 22nd, for Research and Discovery Grand Rounds at that point. So with that, have a great rest of the afternoon.